Good morning, good afternoon, everyone. So today our agenda is, let me share my screen, is as follow. Okay, so Lucas will present some analysis of response strength across time in SLAB 2 sessions. And then I'm planning to present a full solution for Bonsai script for the oddball task that would support all of the variants that we have discussed in the paper. Before we start, I want to give a couple of updates on what we're doing at the island. So last week we ran 2 experiments for SLAB 2 on Wednesday with Andrew Sheldon. And so this was actually a pyramidal cells recording as well as a VIP neuron. So we have those two. The VIP neurons, we actually recorded 2 cells, one superficial and one deep layer, to compare their inputs. So I think that's going to be interesting. We will work to package data hopefully before I leave in mid-July, but I don't know if I can guarantee it at this point. So we'll do our best. But that's good. So we're making progress. And so we deployed the stimulus that we discussed last week with the little extension that you requested, Lucas, at the onset of the orientation block. Okay, why don't you get started, Lucas, and then we can discuss the stimulus after that.
Sure, yeah. I'm really happy you included the extra fluff. This will be the topic of this talk. So just make sure I don't share the share button. Do you see the slides?
Yes.
Okay, perfect. Okay, we're getting better each time.
Yes, thank you.
Okay, great. Okay, so today it's about transient dynamics of control and oddball responses. These terms might be a bit, especially the control might be a bit like odd for some people. What I'm referring to is this idea that we can show the stimulus in different contexts. You know that there's the context where we have the oddball, so stimulus is shown redundant, with many repetitions, or it is deviant, so it's a very rare deviant of this redundant context. And we have the orientation tuning component, which in this sense serves as a random control because the same stimulus occurs without a predictive context in this case. So, and the cool thing is if we can observe the same stimulus under these different conditions, then we can ask questions like, what is the contextual modulation of responses to this stimulus, depending on the predictive context.
Can I ask a quick
yes.
question?
Go ahead.
So my understanding of the benefit of showing these 3 conditions is the following, and correct me if I'm wrong. So if we don't have that control condition, which is, I think, what a lot of oddball paradigms lack, then we might run risk that if the oddball, the deviant, produces a response that's just about as large as the control condition, we might mistake it for an oddball response, but it isn't. Is that correct?
Exactly, yeah.
Thanks.
So it's true, exactly, yeah. So there's this concept of mismatch negativity, right, which is comparing response during, redundant or deviant context. And exactly, one lacks the reference to say this is, above just a simple stimulus response that is released from some form of adaptation. So yeah, exactly. Yeah, so this is an analysis that has been done in the paper by Jordan. Here it's population averages. And you can see that over average of the population, there's a stronger response to the deviant compared to the redundant context. And importantly, the the deviant response is above what you would expect from just a simple, let's say, stimulus response without predictive context. And this is SOMATA. So the question is, do we see something similar for the ROIs in the SLEP2 data? And there's one important thing that one has to do to get conclusive or interesting results here, to focus on responsive ROIs. So these should be, we should only include those ROIs in these population averages that are actually responsive to the stimulus. Otherwise, we will drag down the averages, and we might miss some effects. Responsive here is
sorry to interrupt.
Yeah.
Sorry to interrupt again, but how do you define responsiveness? One pushback may be, well, maybe there are some neurons that only activate to oddballs, and now you're excluding them from your analysis.
Yeah. So to account for that, you take them active. So if they are active during either of the conditions, you include them. So you include them once they have been active during one of the conditions. But you're exactly right. So if a neuron is not responsive during the control, for example, then it's responsive during the oddball because it's enhanced. You have to also check if it's responsive during that oddball. Yes, that's exactly right. So this is what I'm doing here. So I only include responsive ROIs that are active during any condition, and I check that per condition. So I take the baseline per condition. See, is the activity above one standard deviation of the baseline mean, and then I include that unit. Important note here is Jordan uses a higher threshold for SOMA data. I think so. If I use that, I hardly get any responsive units. I guess that's because we look at synapses that are much more variable. So the standard deviation will be much higher, and we have a smaller signal-to-noise ratio. Just a thing to keep in mind. So I used one standard deviation instead of 1.67. Okay.
I hate to jump in again, but Sarah made a really good point that maybe we want to use area under the curve for all of that, because standard deviation is using the assumption of Gaussianity of normal distribution again, which we know neurons don't have, right? So
yeah, that's true.
And it's difficult to come up with a variance measure for median. They're usually awkward. The median is the solution, right? But when it comes to statistics, we smuggle in the normality or the Gaussianity time and again. But if we stick to area under the curve, as Sarah said, then it's independent of assumption of the distribution.
Right. Yeah, I think that's a good idea. This was just, I wanted to replicate the analysis as closely as possible, but you're absolutely right. One could come up with something better here. But I just want to highlight, here it's not really about a thorough statistical test. It's more like an arbitrary threshold that we set. And we should set that so that we get kind of interesting results. But you're absolutely right. The moment we approach this more thoroughly, we should definitely make sure we do not rely on the Gaussian assumption. So if we look at the averages, so what I'm averaging here is this window of interest. I found that the responses occur around 100 milliseconds. So I'm only averaging this window where we have a substantial response. And then I plot the averages here. So this is average over ROI and this window. And you can see that there is a significant drop of activity during the redundant oddball. That is also the case for the 45-degree, probably because it's an overlapping direction. For the 90-degree, we do not see this drop. So we see this depression. We do not see the same strong enhancement that Jordan sees in his data, at least if we do it like this. Now, what I also did was not using ROIs that are active during any condition, but focusing on ROIs that are active during specific conditions to see if how that affects the results. And one interesting thing is that if you choose ROIs that are active already during the first control, so think of it as a synapse coming from a neuron that is selective for that stimulus, or is at least responsive for that stimulus already at the beginning of the experiment. And for those synapses, we clearly see that there's very strong depression, which you can see here. So there's very strong depression occurring for
Lucas?
those. Yes.
You have a question from Nicholas in the chat. What is the x-axis at the bottom? We don't see it.
I saw the legend above.
Yeah.
So,
So yeah,
right.
Yeah. Okay. Right. Yeah. It's the different conditions. Dark gray is the second control, so the control that comes after the oddball. And yeah, so that's this is we just see a strong effect of depression, which makes sense, right? If you want to depress something, it should be active in the beginning. But what's interesting is that we see also strong depression from the first to the second control condition. And that's an effect that Sarah was already seeing in her orientation tuning analysis, that there's somehow an attenuation of responses happening from control 1 to control 2. And I will come to the point how transient dynamics come in there. So this is, if we look at ROIs that are active during the first control. Now what happens if we ask for ROIs that are active during the second control? And here what we see is that those are hardly depressed through the oddball condition, but show stronger deviant response. And interestingly, here we see a clear potentiation. So it seems that there are ROIs that are potentially not active in the beginning, but then through the repeated exposure to the 0-degree stimulus, there are some changes in responsiveness happening. And just to show you one example that's I think is very interesting is this one. So this is responses of a single ROI to the different orientations. And here I always plot two conditions that you see here in the subtitle. So here, for example, oddball versus control 1. And you see that control 1 is gray, redundant is blue, deviant is red. You see that essentially this unit does nothing in the beginning. It's pretty close to baseline the entire time. So nothing interesting happening there. But if we look at control 2 and compare that either to the oddball or the first control response, we see that there's now a response coming up, and it's for the 45-degree, we can nicely see that it has a prominent on response and also an off response, whereas for the oddball here, it has a clear on response, but lacks the off component. So I think that's very interesting. So there's a complex response profile occurring just in the second control. So this is really something that develops over during the experiment. Okay, so to summarize these findings, so for ROIs that are initially responsive for oddball stimuli, we find both contextual modulation, where we find depression and synaptic depletion for standard and 45-degree deviant, potentially also for 90-degree deviant, which could be, because we're looking at populations, there could be some ROIs that are depleted or depressed, and some that are enhanced. So we cannot really see that in this analysis. And it looks like there's long-term depression, pretty much for all stimuli. And I put a question mark here because I will challenge this conclusion in the next part. And for ROIs that become responsive during the experiment, we see that there is mild enhancement for the strong deviant, and we see plasticity in the form of a long-term potentiation for the standard stimulus and 45 degree. Okay, so what I would like to do now is to not only look at blocks per se, but
Lucas?
yes, go ahead.
I think it's maybe a good time. Nicholas had another questions. Maybe you want to express your question on bleaching, Nicholas?
Yes. So here. So yeah, how do you distinguish the effect of photo-bleaching from true attenuation of the synaptic response Since you're, yeah.
Yeah, I mean, so I use the delta F over F data, and I'm assuming there's hardly bleaching. That's of course an effect that could, I mean, there could be a couple of effects affecting responses over time. When I plot them over time, I do not see, let's say, like a gradual decrease in responses. It's more discrete depending on the block that one shows. I will show this in a bit. So my gut feeling says, or I mean, also Kaspar said that that the effect of bleaching in this type of data is it's not such a big problem. I think. And
although you do say that you're noticing long-term depression for all stimuli.
Yeah.
How do we know that photo bleaching is not responsible for that?
Yeah, let's go on. Maybe we can have that discussion in the end after we see the next analysis. It's a good point, and maybe I'm interpreting too much into those data, but yeah, I think we should discuss this after the next slides. Yeah.
Oh, and by during experiment, are you referring to the oddball block?
Yeah. It's saying that in the first, it's responsive during the second control. I mean, there could be units or RRIs that are active during, like, in the beginning and then also in the second control. But typically, what I observed is that units that are, or RRIs that are active in the beginning are so strongly depressed that they do not pass the threshold anymore in the second control. So it looks more like, selecting those units are those that actually are not responsive during first control and most part of the oddball, and then only in the end they show strong response. I will show this in a bit, how that looks like. Yeah, but very good point. Are there more questions to the previous part? Okay. No, I shouldn't have done it. Yeah. Okay. Good. Do you still see my slides?
We see contextual modulation.
Okay, perfect. Great. So let's move on to the temporal evolution of average stimulus responses. So it's a very trivial analysis. It's essentially the same thing as before, but now I'm splitting the blocks, like control 1, oddball, control 2, into sub-blocks to see how responses evolve over the experiment. And this is now shown for ROIs that are responsive during first control. And you can see that, especially for the standard stimulus, responses decrease from the beginning. Then during oddball, the overall level stays roughly constant. And then there is something like a release from the oddball happening. So responses increase slightly. So picking up your point again, Nicholas. So if there was bleaching as the main, driver of depression, one would expect kind of a gradual thing that, consistently moves through all the blocks. But here it looks like a different effect happening, and one sees it a bit in the other stimuli. Yeah, and my interpretation is that this is synaptic depletion. So we start the experiment. We discussed this last time with a completely, let's say, unprepared animal. So it gets the inputs and hasn't seen those before. So there could be very strong response in the beginning, and then there's habituation and synaptic depletion kicking in. And then, of course, during the oddball, there's very strong depletion because we show the same stimulus over and over again. And it looks like that the second control is kind of releasing from this strong depression of the oddball block. If we look at the responses of units that are responsive during the second control, we see a bit of a different dynamic. So we do not see this strong depression during the first control because units are most likely not so active in that one. We have a pretty consistent level of activity during the oddball, and then very rapid, release of inhibition and from depression as well during the second control, and stronger responses in the first oddball. It's very different effect, and yeah, this definitely cannot be explained by synaptic depletion or photobleaching, because the effect is the opposite, right? We see the similar thing for the 45 degree, probably again because of the overlap between the two orientations. And then for the 90 degree, it looks a bit different. Yeah, so just wanted to show you that if we only consider, if we do the same analysis before, but just use the first half and the second half of each block, we see that like this long-term depression effect that we've been seeing in the beginning is just an artifact of, it's just a property of the transient response of these units or RIs. So if we take the first half of the first control, second control, and the first half of the oddball, we see that there's this strong depression from control 1 to control 2, which is because of this transient dynamics, right? But if we look at the second half of each block, we see that the long-term depression effect is gone. So it's essentially going back. It's just looking at this part here and this part here, and they become pretty much comparable again. If we look at the steady state response, if you want, of these blocks, that does not seem to be such a strong long-term depression. And if we do the same thing for the ROIs that are responsive to the second control, the results are hardly affected. So this just shows that these transient dynamics have nothing to do with this type of potentiation. Okay, so how does it fit into the picture I've shown you before? I'd say everything stays the same, but this conclusion that there's long-term depression for ROIs that are initially responsive is not the case. This seems to be rather an effect of synaptic depletion that that leads to apparent long-term depression. And that's because the first control seems high because of initial exposure, so no habituation, and second control appears low because of slow release from synaptic depletion. And for this effect to kick in, the depletion has to be very long, like minutes, in the order of minutes. And this is something that we found in a recent paper uh, in hippocampal neurons or synapses, where the dynamics that analyzed is slow vesicle recovery. So there are multiple stages involved in vesicle recovery. And if you stimulate a synapse for very long, you start depleting these very slow recovery stages. And they take very long to recover in the order of minutes. So it could be that a similar effect is happening here at the cortical synapses. Yeah, so this is essentially the point I wanted to drive home, that there is probably synaptic depletion that leads to this transient dynamics. And we have to be careful to interpret the results. For example, this long-term depression is probably just an effect of this depletion. And I think that fits nicely with the results that Sarah was showing last time, or in a previous meeting, where she found essentially an attenuation of all the orientations, which is in line with synaptic depletion because it's just it affects the overall responsiveness of that synapse. And the cool thing is that with the new stimulus paradigm, we have ways to mitigate this because we add just more control trials, which we can later throw out if they turn out to be transient. And yeah, that's it.
Thanks, Lucas. It's really, really wonderful to see this analysis. I think the synaptic depletion is a valid explanation. I'm just curious if we should follow up with an analysis of the spatial distribution of its effect, because I'm thinking about if it's purely presynaptic, right? You would expect that that will be independent of space allocation of ROI. But if there are, because I think we are potentially well positioned to study intracellular mechanisms that occurs here, and so I wonder if an analysis of the spatial distribution of this effect would rule this out.
Yeah, that's an interesting point. I'm just a bit worried that the problem is that per experiment, we have actually very low number of responsive ROIs in the first place. So, I mean, just to give you the numbers, roughly it's, for example, in one imaging plane, 6 that are responsive for the standard 12. So if those are scattered, spatially, it's really hard to study this effect if so few of the ROIs that we image are actually responsive to the stimulus, because this effect you only see if units are responsive.
Yeah.
It could be that we sample too few ROIs for a proper, to relate nearby ROIs in terms of this behavior. But yeah, that would be a lovely thing to do to rule out, synapse specific effects or something like inhibition locally on a dendritic branch. That's true. Yeah.
But you know where those are, right? We have the ROI mask, so you could just
Yeah,
fairly
I could check. Yeah, it's a good point. I will do that. Yeah, just to figure out if we're lucky and yeah, those responsive ROIs cluster in some
Yeah.
way. Yeah.
I think Nicolas has some code. I think also Sarah to do that. Yeah.
Yeah, also. Yeah, that's good.
Okay. Any questions for Lucas?
I also have I have two bonus slides because someone, I forgot, posted a very interesting preprint last time that does something that we plan to do with the neural pixels. Kind of a similar experiment, but they have some conclusions that I think the insights we got now are really relevant also to their study. Might be, I don't know, if we have 2 more minutes, I could quickly show that how I think our stuff relates to this, but we can also do this another
Is
time.
that the publication that Farzaneh shared last week?
Right, yeah, exactly. Yeah.
I think we should invite these people to present if they are available.
Yeah.
I think that's really relevant. Yeah. Please take a minute.
Okay. Yeah. Yeah. So they do similar stimulus setup. They have a vertical standard and horizontal, and they switch the standard orientation here throughout the experiment. So they also have 2 upper blocks and then 1 control block. Yeah, the stimulation parameters are different. They have longer presentation and blah blah blah. But apart from that, it's quite similar. And what I found interesting is that if they look at the LFP potential, they see so now it's upside down, right? Negative is strong response. The control is in blue, and you see that in the standard context, they have also see a strong depression, and then during deviant condition, they only have a very mild enhancement that's very similar to what we see with these synapses. And I mean, this result is essentially the same if you look at the firing rate. Yeah, so overall, I would say their results are very consistent with what we find for the synapses, which makes sense because probably it's the same neurons, just imaged either at the synapse or the soma. However, there's there's a conclusion that they make, which is the switches in orientation coding depending on stimulus predictability. And so what they do is because they have the same stimulus in these different contexts, they can also compare the response in the same context, but for the 2 stimuli. So they get essentially context-dependent orientation preference. So if you, for example, if you compare this standard with this standard, you get something like how preferential is the response to one of these orientations, but within the same stimulus condition. And they see that this orientation preference depends on the predictive context. So if it's redundant or deviant, for example, for this unit, they see a strong preference for horizontal, whereas in the control, they do not see this. And so they argue that the predictability, stimulus predictability alters this preference. Now, these blocks are only 5 minutes in their case. So they have few trials, they have only 20 trials per stimulus. So overall, the stimuli are short. And so the effects that I showed you, this long-term depression can reach far into this block and substantially affect these results. For example, in this case, for this unit, here the horizontal responses have been stronger, but in the control, the horizontal seems to be depressed actually compared to the standard, right? That's actually very, yeah, I mean, not like weird effect. And I think it's because there's depression going on here, which then reaches far into this control block and gets brings down this response. So I think a lot of their conclusions could actually also be explained by minute-long adaptation effects and potentially also plasticity, so long-term effects that play in here. And I think the lesson we should learn from this is that if we compare if we compare learning or if we want to really investigate contextual modulation, we have to be very careful to dissect these effects with careful analysis and also stimulus design. And yeah, I think we're doing a pretty good job with this already. And yeah, I would love to hear their opinion on this. And yeah, that was the bonus slide.
I'm gonna reach out to these people and try to see if they're interested in presenting in this forum, because I think there's a lot of overlap. I agree. I do want to keep time for the stimulus discussion. Alex, is it a big point you want to make, or? Okay. So let's move on to the next. It's a really important topic related to the stimulus design point you're trying to make. Because we are working very hard to prepare the 2-photon and NeuroPixel sides of those data recordings, and it's important that we converge on the stimulus there. And so I spent a fair amount of time last week preparing the stimulus for those sessions, and so I wanted to show how it's working. In addition, we have talked about having other labs use those stimulus designs. So what I wanted to do now was to show how the bonsai code work, so that we have a recorded understanding of the inner working of the stimulus, so that everybody can make modification if they want to. And we can also discuss the content of the stimulus design, although I feel like that might be too short to do this now. And so I propose we do that next week just before I leave on vacation. And so this way we have that longer discussion on stimulus. Okay, so let me share first the original intent. So when we okay, when we initially review the literature, we made a plan that contained these 4 different contexts, right? So the standard mismatch context, which is what we've been piloting on SLAP for a while now, the sensory motor mismatch context, as well as sequence mismatch, and a jitter context where the duration of the oddball is changed. And the idea was to have the same oddball in all of those different contexts, as well as control blocks that are shared. So we are planning to do 4 different types of session for the oddball block with control blocks that are shared across all of them. So it's actually quite demanding in terms of stimulus coding, because there's a lot of different types of context that need to be taken into account. And so I spent time building that. So let me share what I wanted to do was to share my Windows machine. Let's see if it works here. Okay. Can you see? Oh, you're on the logging screen. Can you see the bonsai thing?
Yes.
Okay, so the way so I'm just going to work this through. Bonsai is fairly sophisticated, so I don't expect everyone to sort of follow immediately. But what I want to do is explain it so that people that goes through the code later on can have this tutorial recorded. So there are several components in the stimulus. The top row here is setting up the window on the screen. The second row here is setting up a little square on the bottom right corner of the screen so that we can monitor timing. Then this row here is used for ending the experiment manually. So you can either trigger the end or press a key on the keyboard so that you can end it. So this is just by pressing the end keyboard of the keyboard. And so that's why it's sent to the workflow output to end the whole experiment. Then this is setting up a folder on the machine where we store the data. It's a default folder. Then this is the setting up the color balance and gamma calibration aspects of the experiment. So it's projecting the stimulus onto a sphere so that you have the same spatial frequency from the mouse perspective, as well as doing coloring. If you want the stimulus to be red, you can do that. So for instance, on SLAP2, we're doing blue stimuli. But on NeuroPixel, we're planning to do gray. Okay, so it's doing that. And then this row here, this whole thing is the most important one of the whole bonsai. And so the way it works is currently is we generate CSV files on the machine that contain all of the stimulus parameters for a block. So let's say, for instance, I open this CSV here. So you see it has a lot of rows that contain all the parameters that are going to be displayed in order. And so in this particular case, you start with contrast 1, diameter 360, a durations that is very short, only a 60 hertz frame, a spatial frequency, a given spatial frequency and a given phase. The beginning here is the motor block. So we have a phase that comes in. And then as you progress through the experiment, you change context. So here we are presenting sequential controls. And so there are longer stimuli with varying orientations. So this is just the parameters of the experiments. And here, this object, the concat object, is taking the rows once they complete it. So this first row is loading a single CSV files that contain all of the stimuli that are in the CSV file for a given context. And I'm going to show you how it's generated later on. So it executes that CSV, and then it moves to the next row, which is the second CSV. So it's fairly, I think it's fairly simple there. So you just have to have as many CSV as you have blocks in your stimulus. And so in this particular case, the first row is the ALBO block, because you remember in our PDF files, we have a figure where we show the ALBO blocks first, followed by all of the controls. So this is the ALBO blocks followed by 4 different types of controls. And so this will be, and you can check these by looking at the enumerate files here object. So you see this is a jitter context, this is a sequential context, this is standard ALBO context, and this is motor ALBO control. So to make this work, I have to do a number of things. So first, I made a custom phase grading sequence object for Bonsai that's share in the, and what I'm showing here is also share on the repo. And so these receive all of the input for the parameters. So that's the main row that you want to understand. And you can add new blocks, but just adding one row to the concat here. So it's fairly easy at this point to make more complex series of blocks. Then this row here is reading the wheel of the mouse and converting it to a temporal phase. So this is what we use for coupling the gratings with the wheel, and it's fed back in. So the temporal phase here is fed back into the first row, because this is the row that can, if we ask to, control the wheel. We ask the mouse to control the wheel. So this block here is going to use it if the CSV says in the phase. So if I open certain CSV, let me show you that part. So if I go to a motor ALBO block here, the CSV for that, you see that the, oops. Oh, this is a control block, sorry. I need to open this one. So there are two types of motor audible blocks. Remember, there's one where the phase is controlled by the mouse. So this column is just saying wheel. So it means the wheel is providing the phase. And for us to allow reloading the previous behavior of a mouse, we have to have this ability in the code. So if you provide the phase in this column, it's going to use it. If you say wheel, then it's going to read it from the mouse. Okay, and so that's this part. So this part here is converting the wheel into the phase of the grading. Then this row here is just doing some path for storing a path of the data. This is saving the result. So essentially, this is saving the stimulus table that just occur. And then this last two things here are very important. They are deactivated at the moment because they talk to the NI card we have on the rig to send pulses every time a frame is flipped. So this allows us to keep track of time. So that's how this bonsai thing work. I also added so and then if you play it, if you play it here, it stopped the stimulus. So you see that's how it looks. So you have drifting grading. So now we are into the first block, and then that's the little corner I mentioned. And anybody here should be able to let me share the other part here. Anybody here should be able to download the bonsai files and run it. So if you go to here, if you go to stimuli list of script, so I generated a little explanation here. So yeah, so there's this generic oddball protocol that I just described to you, and it's in this path on the repo, so you can download it. And there's a page here that explains how it works. There are two pieces to it. One is the bonsai file, and the second one is this Python script that I'm going to explain now. And so this little Python script is located in the code. It should be, oops, sorry. Here, generate experiment CSV. And so what this thing does is you provide a list of parameters that you want to use. I think the code is fairly easy to read. As default parameters, the parameter you want to change, and what it's going to generate CSV files for you, the one to describe. And so those are currently committed to the repo under blocks. And so there is, for instance, if you go to sequentials, you'll find a control CSV, as well as 10 variants of the session. So currently, the simplest way I could implement shuffling with Bonsai in the way that I feel like scale well is I generate a lot of these, and when the sessions start, I just pick one random out of these so that the mouse cannot predict what's going to happen to it. And so any one of these contain all of the trials that a block will occur. And then when the experiment is finished, this file is saved again, except that in the sensory motor cases, the phase is replaced by the actual value. And then you can reload files that are saved by a previous experiment and replay if you desire to do so. So this whole effort is a lot of infrastructure to establish the ability to do all of these things together. Right. So this is what I wanted to show. I know it's a fairly quick, but I wanted to make sure that I have an explanation on recorded of how this work. Are there questions on this? Now, the thing that we probably want to discuss, so now that I'm able to do exactly what we have on the screen now, so the code is ready, I think what we should probably discuss is whether any work that we did with slab should influence how we position the control blocks. And I was hoping that we have that conversation. And does the group feel like this is still a good plan, or is there any things that we think we should consider changing?
I personally, I never worked with any of the other mismatch paradigms, so I would feel more confident to contribute on a case by case kind of if we say like we do sensory motor mismatch next, then yeah, focusing on in general, depends probably, I guess, on how like the exact mismatch paradigm, how one would position those blocks. But I think one pre, one post is in general a good idea.
Oh, you mean the control blocks
Yeah.
before and after?
Mm-hmm.
I mean, that
Yeah.
it takes time, right? Because we have 4 of them.
Yeah, and also for the yeah. And I see. Okay, so you literally want to have all of them in the
Yeah.
same experiment. Oh yeah, okay. Yeah, then that's a different story.
Kareem?
Yeah, so regarding the sensory motor mismatch block, are we changing any of the parameters that relate the visual flow to the running speed, given what we now learn from the standard mismatch condition analysis?
Can you maybe be more specific?
So if I understand from our earlier discussion, there was we were replicating basically the sensory sequence in the sense that the animal is running at an arbitrary speed. The visual flow sometimes follows the animal with the exact one-to-one gain.
Yeah.
Sometimes it's slowed down, sometimes it's sped up, if I remember correctly.
Right
Are we
now,
not changing that?
we are, I mean, we can do things, but right now it's implementing exactly you see on the screen. So 4 different types of oddball. Either the drifting stops, so it essentially coupling is 0, or you have an omission that is presented, or you have 2 alternative orientations.
And we're not allowing anything in between. So as the animal is running, yeah, as the animal is running. So I would suggest we introduce at least some gray gaps in between the switches as the animal is running to distinguish that from the actual dynamics of the speed of the animal. Because the speed of the animal is going to be controlling the temporal progression of the stimulus.
Mm-hmm.
And that will complicate the analysis a little bit because now you have you have to account for the variety of speeds that the animal will have throughout a given trial or block.
Yeah.
So one potential way to have at least some control conditions squeezed within those blocks where you're switching to a different orientation or you're decoupling completely the orientation from the speed, you would probably have some gray screens like the one you have in the standard mismatch here to allow for at least some calibration signals to be collected and compared.
Gray
If that
screen.
makes sense.
So you want to have periods where there's no drifting gratings.
Yes, because if you look at across all conditions, there are periods where there is none, with the exception of this condition. Right?
That's right.
So unless there is a very strong reason for not having one, I would suggest we have one so that we can compare, for example, with those periods where there's no gratings at all.
I think that makes sense to me. So spontaneous activity while the mouse is walking.
Exactly.
Yeah, we just need to figure out how much time we need to allocate to that.
That's, I mean, if anything was learned from the standard mismatch experiment, maybe we should be guided by that.
But I think it could be fairly intuitive to the mouse because it's just going to be like walking through path where they are grading sometimes and no grading sometimes. So it's not going to confuse them. I would expect because it can still be coupled if you're in the dark.
Right.
Yeah. Okay. I mean, it's fairly easy to implement. So maybe as much time as there is omission. I can make a proposal and then we discuss it on the forum.
Okay.
Any other? So I would like to propose that next week, for the last meeting before I'm away, we discuss these in more depth because I think it's an important. Does that make sense?
Yeah, makes sense. Just one high-level question, Tom. So the idea is, so I'm not up to date anymore with the paper. So the idea was to have all the mismatches in a single experiment, or only the controls, and then we have individual experiments per
Yeah.
mismatch.
So you see that figure 11 here?
Yeah.
So the intent is to present only one of the elbow block for the session and then share the controls. And so then the one day you do one of the context and the next day you go back to the same animal and change context. In 2Photon, that would allow you to record the same cell. In NeurPixels, that will likely be different cells, but we'll do a new insertions.
But each session has both control and the context, or it's the control comes at a later point.
So you see here, this is chronic recordings where we just record one context at a time, and then
Right.
that will be this block, and then at the end of the session we'll have all the control blocks in series.
What's the rationale to have the control, like also the other controls in that session? Wouldn't it make more sense to just have the control that matches the mismatch
Yeah.
condition in that individual session?
The intent was to use the control blocks to relate the different session with one another.
Okay. Okay.
Because you're not sharing the elbow block.
Is it crucial to relate them? Yeah, that was one of the targets, right?
Yes, it's important because we want to compare the responses change during the elbow block. So having this control shared allow for like first normalization of each context to each control, and then comparing across elbow blocks. Because if there is anything that is different during that day, you have the control.
I see. Okay. Yeah, thanks.
So why don't we regroup next week to discuss? I propose we have a less structured discussion, but focus on iterating this design, leveraging what we've learned on SLAP. Does that make sense? Because I think we've learned a fair amount, and it's important that we do this right. Sounds good? Okay.
Yeah.
Thanks, everyone.
Thanks.